So, first of all, thank you, Michael, for inviting me to this forum and to the society. I'll go. The main topic of my time with you is to share these very basic and I think simple and straightforward ideas of how we could go from the FAIR principles that are very much focused on data into something equivalent that could take care of increasing the quality of representation of knowledge. So this is my team. It's a part of my team in Mexico. There are some people that are missing, and Claire is part of Elixir. She's a former student of mine with Jacques Van Helden in France. So, so I think, all this revolution of AI is inviting us to increase the explicit clarity, precision, assessment, best practices all over, and quality of knowledge. As I said, the FAIR principles is sort of the start point we are all know about. And but biodata resources, include also knowledge or facts. So the conceptual framework is this one is my, I've been working for 30 years on knowledge representation, on addressing, gathering, curating knowledge of transcriptional regulation in E. coli. So it's a small model organism, but has the sort of the best known, I think, transcriptional regulation in terms of details of different levels of mechanism, physiology, and so on. So based on that experience, through the years, I've come up to these simple ideas that any type of knowledge, not only scientific knowledge, in principle has an evidence, right? In our case, of course, in science, the corresponding sources, references, an explicit level of confidence level, right? Knowledge is not equally confident in different domains. And in our case, it depends very much on the methods that we are using behind supporting every piece of knowledge. And there's no isolated piece of knowledge, right? Whatever strange idea you have, you can come up to the logical opposite, and things are not isolated. So these are, just to briefly say the proposal of the essential criteria of knowledge of quality representation version 1.0. If following this idea of clarity, I suggest this definition of wisdom, which this type of triangles are common to people from data to information, knowledge, and eventually wisdom. There are no precise definitions of the difference between one and the next one. So I propose this definition of wisdom to be the one is the knowledge that enables us to live at peace with ourselves, with others in society, and with the planet, right? And of course, well, the other thing is biology is sort of the major fountain of diversity in the universe. It's really an energy for creating diversity. So based on this, I was just looking last night to this statement of the Society of Neutrality, the vision of ISMB to lead the field of computational biology to advance science and all society. So based on that observation, I just added this, what you can read there. I think humanity urgently needs much more wisdom than intelligence. Okay, so I come back to the main set of ideas of my talk. It's very easy to say, okay, we need everything, we need evidence, but it's easy to identify levels of increased quality of representation, for instance, in terms of evidence. First, we can say we want to have every object in a database to have at least one evidence and one source. And you can improve that to have not only the objects, but also the features. Sometimes, like for instance, I will be giving, of course, examples of gene regulation. So for instance, a promoter is a complex object that has in bacteria different sigma factors and these are different experiments for features of that object. So eventually we think of not only one object, but every feature to have its own evidence and corresponding source. To go and quote the original publications and not only, reviews or papers that mention the given piece of knowledge, but are not the initial source of that piece of knowledge. To have a one-to-one relationship between one evidence and the associated source. Sometimes, sometimes we can have, a collection of evidence and a collection of references, and we don't easily know which go with which one. And then, of course, any other implementation you can think about that will enhance the directedness between a piece of knowledge and the corresponding source. So as an example, that's in terms of, the first criteria. And then in terms of confidence level, as I was saying, it's not the same confidence we have of the second law of thermodynamics with the prediction of weather, right, for tomorrow. So in RegulonDB, we started with the very simple idea that we will distinguish between strong and weak evidence, where strong is the one that is supported by experiment that provides physical evidence for the existence of the object of the interaction, and otherwise it's a weak one. So I'm going to use examples that we have been implemented in this Regulon database. As I said, we are also the ones that feed all curation of gene regulation to ECOPsych. And ECOPsych is sort of the foundational model for Peter to go and expand it to biopsych and other microbes and so on. So for instance, we have added source sentences in our collection where source sentence is the precise extract sentences in a given paper that best describes the given object or interaction or so. It's something we have started relatively recently. And in terms of confidence level, we started initially with adding all sorts of weak and strong evidence, but initially they were not additive. So we came to realize that some objects have multiple weak evidence. So with Verena Weiss is a former, a German colleague that we were both postdocs at MIT years ago. We worked together and we defined what are the criteria for identifying independent types of evidence, right? Whenever you have different methods that provide. How do you identify independent methods based on they provide sources of different errors, different assumptions, maybe different chemicals, right? So based on that, we started. I'm now showing you the updated criteria for, especially with this tsunami of high throughput data. The classification we have of the methods in terms of identifying regulatory interactions. So you have what we call the classic, binding in vivo or binding in vitro of purified proteins. So these are the only ones that have per se strong evidence. Otherwise, cellular extracts are weak because they could be indirect interactions. All high throughput, we consider them to be weak, and in order to do this, we had to go from the eco ontology of evidence to go down into specifying the methods. ECO doesn't specify the methods, and we needed to do that to, so all ChIP-seq belong to one group. So these methods here in one of these boxes means they are not independent, all of them, right? But one box to the next one is we consider them to be independent evidence types. And then we have the non-experimental ones, and we have, based on this, sort of built up an algebra. I call it an algebra of which ones can combine, and the list of all these are in RegulonDB. You can see there here S for strong, weak, and confident. And of course, this is our construction, and all the information is detailed so that anyone can make up, if they don't agree with our algebra, so to speak, they can make up their own. Now, when we started with these ideas years ago, ECOPsych the advisors weren't happy about me proposing weak and strong, because they said, oh no, authors, we don't like that. You assign the label of weak evidence to, right? But so ECOPsych doesn't have this, but we implemented this in RegulonDB. And one of the interesting things is that this confidence level has been very useful for benchmarking a lot of new high throughput methods, as I'm showing you here a partial list of, GCLX, ChipSec, ChipXO, EXO. So many people have always compared the results to what is known, described in RevLonDB. And this was also essential because we were wondering about the combination of what might become a less and less frequent classic experiments with the huge amounts of high throughput data. Context is the other one that I think it's a universal property of any type of knowledge. And I kind of think of, this is just yet to be, sort of ideas to be implemented. So we have, external context that goes into metadata, authors, and labs, and replicates, and so on. And then internal context in the sense that properties that are inherent to knowledge, given the structure of knowledge, of biological knowledge. So these are properties that are true for irrespective of which database you are, gathering data of whatever piece of information. So in my perspective as a biologist, the context of gene regulation is that this is the machinery that enables the cells, on the one hand, to enable one genotype to have multiple phenotypes, which is foundation of biology. And then regulatory networks are the mechanism for cells to be able to sense the environment, external or internal environment, and modify, go and knock the door to the genome and modify the expression of genes. So this concept is formalizing what we call the genetic sensory response unit. You can see there on the diagram, the four components: the signal, the transduction, the regulatory switch, and then the response back to whatever genes have been modified. And we can see here in terms of, so that's the one of the contexts of knowledge of gene regulation. And of course, you have is part of structure of promoters being part of transcription units, being part of operons, and so on. And this sort of raises the question of whether we can eventually order knowledge, not only, and we have, I'm showing you here some, we are implementing, of course, AI, prompts to try to order sentences from papers in terms of these precise different components of a gensor unit. So we'd like to do that, of course, to the whole collection. And so that's on level 1 of version 1, so to say, of quality of knowledge representation. But there are other properties that I think are also universal, like the one of any piece of knowledge can be described at different levels of detail. So I like this, I love language, natural language. And it's amazing when you think of, if you say microorganisms live in environments subject to rapid changes, you're also saying all those, shorter sentences, if you just eliminate one word at a time, no editing, just eliminating words, you can come up to microorganisms live, and you can even go higher and say organisms live and life is made, lives in organisms, right? There's something like that. So this is a natural structure of knowledge, of any type of knowledge. And so I was, before even ChatGPT and all that, we were already in my lab, manually got a group of curators and myself into identifying 5 different levels of detail, from highly general to categories, to plurals, to individual objects, and each of them, the sentence is giving generalities or specificities. So we play with this idea, and a while ago, to connect this with an application, a while ago with Pascal Claudette from the Gene Ontology and colleague experimental microbiologist, I made the observation that if you go and extract all the gene ontologies that are required for annotating the whole collection of genomes in E. coli, you come up with 19 highest level categories. I'm showing you here some few, like metabolic process, localization, cellular process. And if you think you are the teacher, you're going to teach the essentials of microbiology to students, these are useless, right? You have to go from, so these are not capturing the general principles of microbiology, right? And I think that is an interesting absence of high level of generalities in all sources of genomics, I bet, right? And so, it's something like I don't know the word in this Chinese feng shui or whatever. Things have a place, right? Knowledge is a structure, and knowledge has a place in terms of all these contexts. So the dream is, how could we connect data with all these general statements, right? And now that I step by at Birmingham with colleagues, experts in gene regulation, we are going to start the project of precisely, ordering the knowledge of Regulon DB, starting from, experts getting into, and also from experts reviews, extracting those general statements. And so I finish with this idea that in biology, our instrument to generate knowledge and to look far away is precisely, bioinformatics and AI are instruments that use knowledge. The clear example is multiple sequence alignment, right? You do that, and then from there, you can go and extrapolate knowledge from one organism to others, right? Sequence conservation, and right? So if we really, we know that biology is extremely complex at many different levels of detail, of working of systems, multidimensional. If we really want to have now with AI to construct sort of the whole of biology, we need to, we better align knowledge or organize knowledge in a way that it will increase the sharpness, the precision of our instrument in the long term. So in terms of the social implication, it's very clear. I met well, I don't know if you know that book from a colleague here in Edinburgh, of all about this misinformation and also the lack of, we know about the lack of reproducibility and problems like that in natural science. I'm showing you here a curator got into reading 600 papers to extract. We eventually developed what is called the microbial condition ontology, MCO, and based on essential knowledge of microbiology from Fred Neihardt's book, we got into the collection of properties necessary and sufficient to reproduce an experiment, and one of them, essential, is growth rate, which, as you can see, is missing in the description of papers, of 600 papers. So that shows, it's just one example of what we all know about. So, yeah, I just played yesterday with the same type of minimizing the sentences. Thank you. I just finish with saying, these statements, my major interest is to go and work with whoever is interested and can contribute, so that we can not just, I could just go and make a paper of this, but I'm much more interested in saying, is there a way that we can, as a community, work into constructing and implementing this, quality of knowledge equivalent to the FAIR principles, starting with whatever, 2-3 databases or people in the theoretical component of knowledge representation. Yeah.
Thank you. We have time for a quick question.
So this is like truly ambitious in a very good way, and I'm wondering if you think you see this sort of plugging into some things that might already be existing or emerging, like things that come to mind are like the big history movement, or things like cliodynamics, where there's like serious effort to like look at historical trends and project forward into like what could be like futures studies, things like that. Or is this really targeted for life sciences without really trying to encompass other things?
Yeah, thank you. I am much more interested in, I guess, starting in life sciences as not only, a single, isolated idea, but to go into implementing, things like organizing metadata or really finding the location in the ecosystem of annotation, curation, representation of the tools that are mostly used, and that how we can, incorporate this notion of explicit context is sometimes present there in many resources, but it's not explicitly called, context. And then we could just use that as another, interoperability mechanism.
Thank you.
Thank you.